goats are considered as the grazing animals. They are more prone to the poisonings, and especially the plant poisoning. The ruminants are vulnerable to the hydrocyanic acid poisoning. The sources of poisoning, cyanogenic plant, that is the main source of hydrocyanic acid poisoning, especially sorghum. Sorghum accumulates HCN more, and therefore the feeding of sorghum can lead to a hydrocyanic acid poisoning. Then Acacia leucophilia, source of HCN poisoning, in my opinion, to the goats. This is the most important source. Then linseed meal, that is sometimes, or linseed cake sometimes may cause. There are hundreds of plants and grasses which can cause hydrocyanic acid poisoning in goats. But just I have given a list of 3 here, because which are most commonly responsible for HCN poisoning. See, the jowar fodder is normally being fed to the animals, but it doesn't cause, the hydrocyanic acid poisoning. If it is well grown and if a mature fodder is being fed, no problem of this hydrocyanic acid poisoning. But immature or very young and rapidly growing jowar fodder, if it is fed, then the HCN poisoning may result because of high levels of HCN. Fresh forages, they are rich in HCN, they contain high levels of HCN, and therefore they are responsible for the HCN poisoning. The dry forages or the silage or hay doesn't contain HCN, because HCN is volatile and it gets dissipated while the process of drying. The new sorghum growth following drought or frost, this is the worst situation. Particular area receives rains following a spell of drought. And that leads to a rapid growth of, the jowar fodder, or that jowar contains a dangerously high amounts of HCN. Then trampling, cutting, crushing, chewing, and wilting of the plant leaves may cause a release of the HCN. So these are some of the factors. Then why ruminants are vulnerable? Why are in other animals, HCN poisoning doesn't occur. Cyanide poison can occur in any animal species, as such cyanide poisoning. But hydrocyanic acid poisoning occurs only in ruminants. Why? Because HCN is liberated in the rumen from the cyanogenic glucosides or glycosides present in the fodder. So ruminal microflora, they have got ability to hydrolyze and release HCN from the cyanogenic glycosides. These are the plants which are responsible for HCN poisoning. First one is jowar. Actually, the very young jowar fodder is responsible, even a sorghum or sudaran grass, then velvet grass, Indian grass, Johnson grass. These are the various grasses, particularly immature and rapidly growing grasses. They are responsible for the HCN poisoning. Linseed cake sometimes. But here I would like to highlight these 2. That Acacia leucophilia. This is the you can say a bubble tree. It's a bubble tree which is found in many, you can see the drought. It is found in the drought, widely spread in the drought-prone area. It survives in the drought, and that too on a poor soil. And generally, that the sheep and goats, the population is very high in a drought-prone area. It's more in our drought-prone area where this plant, this particular tree is more common. And I have seen number of outbreaks of HCN poisoning during summer season in goats following ingestion of parts of and the seeds of this Acacia leucophilia. During the summer, the animals, they consume goats and even a seed they consume. And immediately after consumption, you see, this is a dry type of pores when they consume. And when they drink the water, immediately they start showing the signs of HCN poisoning and they die. So I have seen number of deaths due to this. Therefore, I would like to share this information. The cyanide ions have a great affinity to ferric ion. See, the HCN from HCN cyanide ions, they are being released in the rumen, and those are being absorbed into the bloodstream. And these circulating cyanide ions, they have got a great affinity to the ferric ion of the cytochrome oxidase. You all know that cytochrome oxidase is the enzyme required for a cellular respiration. It is required for a cellular respiration. And this particular enzyme gets inhibited. So because of formation of cytochrome cyanide complex. So this becomes inactive, and therefore cellular respiration will not occur, and that results into a cytotoxic anoxia. This is the cause of death. Here in cytotoxic anoxia, oxygen present in the blood cannot be utilized. Oxygen is present in the blood. Actually, oxygen is sufficient, but the tissues or the cells, they cannot utilize oxygen because of inhibition of the cytochrome oxidase enzyme. The typical clinical signs which are seen in the initial stage, usually this HCN poisoning is very acute and the onset is within half an hour. And many times death occurs within half an hour to 1 hour. Many times we don't get chance to treat the animals. This is a very poor acute to acute type of a poisoning. Initially, there is a bloat. In a milder type of the cases, the signs of bloat are evident and the cases are being reported. Then there is a labored breathing. There is difficulty in breathing because of anoxia. There is a shivering, tremors, and the animals show incoordination because of this anoxia. Animal is restless, may fall down, show opisthostonus, that is upward deviation of the neck, and the abnormal movements of the eyeball, convulsions, and death within half an hour. But here I would like to share a most typical sign of HCN poisoning. That is bright red mucosa and bright red colored blood. Poisoning without performing a picoret test or analysis under the field condition by seeing the color of blood, and mostly by seeing the color of, you can see the conjunctiva. All the mucous membranes, they are bright red. They are like arterial blood. Therefore, the word bright has been used. So that is typical. And if you remove the blood, you can very well see that it is a bright red. HCN poisoning is very acute in nature, and usually the onset is within 30 to 40 minutes after ingestion of the plants rich in cyanogenic glycosides. And even the course of the poisoning is also very short, and death usually occurs within half an hour to 1 hour. Among the clinical signs, the bloat is the initial sign. And bright red mucosa and bright red colored blood is typical of HCN poisoning. One can very well diagnose a case just by seeing the color of mucous membrane. It is just like a color of a blood. And because of anoxia, there is a difficulty in breathing, as well as animal will exhibit the nervous signs because of anoxia. And in dead animals, if you cut open the rumen, then you will get a bitter almond smell. How to treat the cases of HCN poisoning? First of all, you have to remove the source of poisoning, stop feeding the suspected feed material, and this antidote is a combination of sodium nitrite 1 gram plus sodium thiosulfate 3 gram, that is to be dissolved in a 50 ml of distilled water, and that is to be given intravenously. Even if one can repeat it after 1 to 2 hours if required. And sodium thiosulfate can be a 10 gram of sodium thiosulfate can be given by oral route. If a sodium nitrate is not available, in that case, even we can use sodium thiosulfate alone, but this is the best combination. So we can also use the sodium thiosulfate alone at the rate of 65 to 66 milligram per kg body weight. Regarding the symptomatic treatment, we can give anti-bloat drugs, carbonatives to reduce the diphtheria, corticosteroids to prevent the shock, and even fluid therapy can be given to the animals. As in mild to mild cases, they can recover within 2 to 3 hours, even if this antidote is not given. But you see, the severe cases, moderate cases, they need antidote. Then second is the nitrate poisoning. This is generally we pay more attention to the HCN poisoning But this is one of the neglected poisoning, but it is occurring under the field conditions. Again, in ruminants, nitrate poisoning occurs only in case of the ruminants. Nitrate poisoning can occur in all animals, including ruminants and simple stomach animals. But here we are more concerned about nitrate poisoning in case of the ruminants. And among the ruminants, again, cattle and buffaloes, they are more prone, mainly because of their nature of the diet. We are giving them fodder, and sheep and goats are grazing animals. Accidental ingestion of fertilizers containing nitrate. So very commonly, the fertilizers containing NPK or ammonia, this N and phosphorus P, they are being used in the agriculture. And therefore, accidental ingestion of these fertilizers by sheep and goat can lead to this nitrate poisoning. The most important is nitrate-rich plants. See, it has been clearly mentioned that each and every plant can accumulate the nitrate. But when we compare the leguminous with non-leguminous or cereals, then cereals, they accumulate more nitrate as compared to the legumes. And therefore, leguminous plants are not hazardous from this nitrate poisoning point of view. Therefore, you see, the only cereals, cereal grasses and cereal forages, all cereal forages. And maize fodder is one of the important source of nitrate poisoning. When we compare it with the jowar, bajra, and wheat, or maybe a paddy, the maize accumulates more nitrate, and therefore maize fodder, you see, is responsible for a nitrate poisoning if it is grown on a nitrate-rich soil, heavily fertilized soil. If its growth is stunted, again the rapid growth following rains. So these are the conditions that creates a very conducive atmosphere for the accumulation of the nitrates. Eating of a nitrate-rich maize can lead to, you can say, the nitrate poisoning. Then third one is water from the deep wells. Actually, the well water is comparatively safe. River water is more safe. Well water comparatively safe, but borewell water is dangerous. And sometimes you may get the incidence of a poisoning in maybe in a sheep, goat, or a cattle after drinking of water from a deep, you can say, the bore wells. Nitrate actually is present in most of the plants, maybe in a small quantity, but that nitrate, when the nitrate present in the fodder is being converted to nitrite, and that nitrite is converted to ammonia. So hydrolyzed to the ammonia. So the ammonia is the end product of nitrate. And this ammonia is trapped by the ruminal microbes and converted into proteins. If a nitrate is small in quantity, then definitely there will not be a poisoning. It will not be, you see, cause any harm to the animal. Instead, those nitrates will be utilized for protein synthesis by the ruminal microflora. But when quantity of nitrate is more, in that case, nitrite will not be converted to ammonia. So nitrate is rapidly converted to nitrite, but nitrite may not be fully converted to ammonia, and therefore there is accumulation of the nitrite in the rumen, and that nitrite is being absorbed from the rumen. And when that nitrite goes into the blood, it acts as an oxidant. It acts as an oxidant, and it causes oxidation of hemoglobin, and we call it as a methemoglobin. Normally, it's oxyhemoglobin that is being converted to methemoglobin. This is oxidized hemoglobin. And this hemoglobin is not having any capacity to carry the oxygen. So methemoglobin causes anoxia. It causes anemic anoxia. Actually, the blood hemoglobin is normal, but that hemoglobin is unable to, you can say, carry the oxygen, and therefore it is called as anemic anoxia. See, when nitrite is ingested by animals, it will lead to nitrite poisoning. Nitrate also is converted to nitrite, and finally it's a nitrite poisoning. So what are the clinical signs observed in nitrite poisoning? The first sign is the salivation. In some animals, there is a diarrhea and colic. If nitrate is the source, usually there is a diarrhea and there is a colic. If nitrite is consumed as such, in that case, the signs of colic and diarrhea may not be evident. But most commonly, it's a nitrate is the source. And therefore, you will find a salivation, colic, diarrhea. These are the GI type of signs. And there is a dyspnea because of, you can say, the methemoglobin. And the most typical finding is a brownish mucosa. Here you can see the brownish mucosa. Those are very typical brownish mucosa, and the blood is chocolate colored. If you prick the jugular vein or maybe any other vein, the blood, whatever coming out, it is a chocolate colored blood. Then the muscle tremors, staggering gait, convulsions, and death within 2 to 6 hours. So the course is comparatively longer than the HCN poisoning. Here, usually death occurs. In case of a purely a nitrate poisoning, it takes 4 to 6 hours. If it is a nitrite and if the quantity of the nitrate consumed is also very big, in that case, death will occur within 2 to 3 hours. Otherwise, it takes 5 to 6 hours for the, I can say, the casualty to occur. This is a typical chocolate colored blood. On postmortem examination, you can see the brownish discoloration of the lungs, and you see all organs, they show the brownish color. So they are stained with the methemoglobin. They are stained with the methemoglobin. Methemoglobin is brownish in color. How to treat the cases of nitrate poisoning? First thing, we'll have to avoid and we'll have to control by removing the source of poison. So if you suspect a deep bore well water, don't, allow the animal to drink. Fodder also, you'll have to change. Immediately you'll have to change. And for treatment purpose, the methylene blue is the drug of choice. That is to be given at the dose rate of 4.4 to 8.8 milligram per kg body weight intravenously as a 1% solution. That means you'll have to dissolve 1 gram of methylene blue in 100 ml of water, and you'll have to give it at the rate of 4.4 to 8.8 milligram per kg body weight. So even 4.4 is good. 8.8 is also good. But there is a problem of availability of methylene blue under the field conditions. And therefore, the alternative that can be used, no doubt it is not effective as a methylene blue, but definitely under field condition, ascorbic acid is available. So it is available in a 5 ml ampoule containing 500 milligram of ascorbic acid, and that can be very well used at the dose rate of 15 to 20 milligram per kg body weight intravenously. As far as the supportive treatment is concerned, you can give a saline purgatives and oral antibiotics. Generally, I recommend oral antibiotics, because if you give antibiotics, then the conversion of nitrate to nitrite will be stopped. So therefore, oral antibiotics will be better. Oxygen therapy, here, actually it has been mentioned in the book, but I don't think it will be of use, because the hemoglobin is oxidized. There is no hemoglobin for carrying the oxygen. So this oxygen therapy may not work. Blood transfusion will work to some extent, but already the quantity of blood is sufficient. And if you give additional blood, then that will cause you can say the overloading of the circulation. So these treatments are of little value or no value as such in case of nitrate poisoning. The magic treatment is ascorbic acid or methylene blue. If you give methylene blue within 1 hour, half an hour to 1 hour, within half an hour, that dyspneic animal will start respiring very easily. And within 1 hour or 2 hours, it will become a normal. So dramatic recovery is there after giving either methylene blue or ascorbic. So just like giving calcium in a case of milk fever. So such a magic is there, no need of other treatment also. Then the third is oxalate poisoning, which is also occurring under field conditions. And it is commonly seen in ruminants. Actually, ruminants are considered less vulnerable as compared to the simple stomach animals, because in the ruminants, the ruminal microbes, they can degrade this oxalic acid. But even then, their dietary pattern is responsible for their increased susceptibility. When the animals, they are maintained on on a oxalate-rich plants or diet, then the oxalate poisoning will develop. The forages containing oxalates greater than 10%, they are dangerous. Only if their oxalate is in a small quantity, it will not affect. So there are many plants which accumulates the oxalates. And among those, in India, this Anagallis arvensis, in Marathi we call it as a dor kakda or a fuligavat. See, this is a very beautiful, you can say, the plant, I will say. And it grows during Rabi season. It grows during Rabi season, generally in November, December, January, and that too in a Rabi crops. It is having you see the leaves, they are very much shining, the flowers, they are very beautiful, and it is very palatable, liked by the animals, and they are in control. And even a very small quantity of this anagalis, ingestion of a small quantity of anagalis arvensis can lead to oxalate poisoning. The others, they can oxalis species and amaranthus and Citaria species, they can also accumulate. Even sugarcane tops, they are blamed for accumulation of oxalate, but the quantity of oxalate is less. That may not be sufficient to cause, you can say, the oxalic acid poisoning in a large way. And there is no question of feeding of sugarcane tops to the goats. That may be the problem in case of cattle and buffalo. The oxalates actually, they chelate the calcium and form insoluble complex with the calcium oxalates. And these calcium oxalates crystals, they block the renal tubules and they cause a renal failure. Even the chelation of the calcium will lead to a failure of blood clotting mechanism. These are the two important effects of the oxalate poisoning: renal failure and failure to clot blood. The clinical signs, they include the severe anorexia and weakness and ruminal atony. Here, I would like to share, I was unable to detect any case of oxalate poisoning in a sheep and goat. I've seen several cases of oxalate poisoning in the cattle and even in the buffaloes, but not in case of sheep and goat. But this poisoning can occur. Ruminants are vulnerable, they are susceptible, and this poisoning can occur. Anorexia, weakness, ruminal atony, constipation is typical, and sometimes this hematuria may develop red urine because of, you can see, the oxalate crystals causing damage to the renal tubules. Liguria is observed. There may be a dribbling of urine and edematous swelling on perineum. In cattle, we have mainly seen the edematous swelling over this perineal region. So we will mark with protrusion of the anus and straining while defecation, and there is a swelling and edema of you can vulva and anal region. So this is very typical of renal failure cases in case of the cattle and buffalo. So we have used this line of treatment in case of the large animal. That's I'm sharing with you the fluid therapy. We are giving maybe a D5. We are using antibiotics to prevent the secondary bacterial infections. Already there is damage to the renal damage is there. Diuretics we are using in case of edema. They are useful you can see maintaining the blood flow to the kidneys and urine flow, and that helps in prevention of further renal failure. Then B complex to regenerate the animal. Calcium borogluconate, you can give it in a small quantity. The dose which we are using in cows, that can be very well used, 20 to 25 ml, that can be given by IV route daily. Again, there are two schools of thought whether we should go for a calcium, because if a free oxalate is present in the blood, that will be combined with the calcium, and again the calcium oxalate crystals are formed. But practically speaking, and we have seen this calcium borogluconate by IV route hastened, and it helps us in the recovery by improving the GI motility and everything. So lime water can be very well used. That is to be prepared 1 kg of calcium hydroxide of lime is to be mixed with the 9 liters of water, and that is to be soaked in you can see the water for our dissolved thoroughly. It is to be dissolved, and it is to be kept stand still for overnight, and then we'll have to take a supernitrate. We call it as a lime water, and that lime water can be given to the animals thrice a day. Maybe for a goat, 50 to 100 ml of lime water can be given thrice a day. Laxatives, they are to be given to remove the to correct the constipation, which is typical of this disease. But if you are giving calcium, then the tonicity will improve, and the defecation will start. Probiotics, they are to be used. Prebiotics and probiotics, they are very much helpful. Fresh rumen curd helps in restoring the appetite of the animal. And treatment needs to be continued for 5 to 7 days. Usually 5 days treatment is required. Sometimes 7 days, 9 days, 12 days treatment is also required. But generally, the cases which are to be recovered, which are to get recovered, they recover within 5 days. The cases which are not responding to the treatment within 5 days, the prognosis becomes unfavorable in those cases.